And let me welcome our second speaker in this panel. On behalf of Professor Janusz Bujnicki, we have Dr. Tomasz Wirecki, who will talk about the second project, which is from RNA structure prediction to drugging the genomic RNA of SARS-CoV-2 with small molecules. Tomasz Wirecki, the floor is yours. You have half an hour.
Welcome everybody to our presentation. So actually, on behalf of Professor Janusz Bujnicki, I will be presenting, but also my colleague from the lab, Dr. Filip Stefaniak, will add something at the end of the presentation. So first of all, I wanted to thank for the opportunity to work with the LUMI Supercomputer. This is super exciting for us, and actually we hope and we believe that thanks to that we will be able to progress with our studies on SARS-CoV-2 structures and possible druggable or possible drugs findings. So now I will start with a bit of background on what actually RNA is and what programs do we have to simulate it and what can we achieve with that. So first of all, RNA is a biomacromolecule made from nucleotides, and for it to have functions in an organism, it needs to form very complex 3D structures. So most of people know RNA, that there are only messenger RNAs that are used for creating creating proteins, but actually RNAs do have much more functions, and for them to function, they need a spatial structure. So here you can see different roles of RNA in biological processes. So despite the one that I mentioned of mRNA, they can regulate gene expression. They can interact with proteins, with DNA. They are responsible for ribosome assembly. They also take part in splicing and then creating different variants of a protein, etc., etc. So for that, if we want to know the function of an RNA, we actually want to know its 3D structure. Also, another fundamental thing that influences the work of an RNA is that it is capable of interacting with small molecules that we call ligands. So as you can see in the image here, that going from sequence, the RNA folds into 3D structure, but once the small molecule ligand approaches the RNA, it can change its structure, meaning it will work somehow like a switch. So we may assume that in the form without a ligand, RNA is inactive, but when the ligand is present, the RNA starts to be active and play its role. So this was shown, for example, for HIV TAR RNA, whereas you can see the structures without ligand and the structures with ligand are different. And you can see the change of the structure when the ligand approaches RNA. And also, this is an important thing in a frame shift element of HIV viruses. So what are the questions that we want to answer with our studies? So how actually the 3D structure of an RNA is encoded in the sequence? The sequence, the RNAs basically consist of four basic nucleotides, GUAC, and they are able to form very complex structures. So we want to know how those structures are encoded in the sequence itself. We also want to know how the change of state or the shape shift happens in RNAs. Then this can be done in many ways, not only by ligands but also by temperature change and other mechanisms. How do RNAs actually recognize other molecules and how they interact with each other? So those are the big questions that many of people doing research on RNA are asking. And what are our approaches to answer those questions? So we basically collect the existing experimental data. We also do wet lab experiments in our lab. We then develop knowledge-based methods, computational methods for simulations and predicting structure. Then we can use those methods to predict the structures of RNA, either during the test for known structures that we can find in PDBs, and later if we know that the methods work correctly, we do predictions of unknown RNAs. Then for validation, we are able to test all our predictions experimentally in our lab, and then improve our methods. And from all those data that we gather, we derive general principles governing the structural aspects of an RNA. As I mentioned before, we are able to do experiments and incorporate the experimental data in our computational workflows. And here, for example, we have a set of experiments that we are able to incorporate in our predictions, like X-ray crystallography, electron microscopy, small angle scattering, and chemical probing. So all those experiments give us some hints about the structure, starting from the top. So X-ray crystallography and electron microscopy can actually show us the actual structure of an RNA in a given state. And we need to remember that in a living organism, the structure of RNA is not just one state, this is an ensemble of states. Whereas actually the best we can do experimentally is to get crystal or electron microscopy structures, but this is very, very time consuming, very difficult, and of course very costly. From small angle scattering, we can get the overall shape of the RNA, which is basically lower resolution experiment, while from chemical probing, we can get information about the state of specific nucleotides in the RNA sequence, and those states correspond mostly to the fact if the nucleotide is interacting with something or not. So from this, we mostly derive the secondary structure of an RNA. And if we want to actually simulate an RNA or a macromolecule, there are several approaches. So looking from the bottom, so this is Greek science approach, where we start from the very principles of quantum mechanics, and then we go a bit lower resolution to molecular dynamics simulations, but those are of course super costly computationally, and are very difficult to simulate a span of life of RNA in a longer time. The other approach is Babylonian science, where we know everything, and from that we get information like from experimental data. We are able to do template-based modeling. So if we want to have a template of one RNA and we know that there is a homologue that we are studying a homologue of this RNA, we can do template-based modeling, so changing the fragments of existing RNA. And what happens in the middle between of the two approaches is actually what we are doing. So we are using a coarse-grained approach where we lose some information, but then we are able to do the simulations in a faster time, getting the rough structure of a macromolecule in reduced representation to speed up the calculations. So for this purpose, we derived a methodology called SimRNA that is used for simulation of folding, unfolding, ligand binding, protein interactions with RNA, where we represent RNA in a coarse-grained representation, which means we don't simulate the movements of all atoms in an RNA structure. We represent each nucleotide with 5 pseudo-atoms. So three pseudo-atoms are for the base, and 2 atoms are for the backbone of RNA, which gives us 5 atoms per nucleotide. Then we use Monte Carlo moves for specific nucleotides and run the simulation of folding, for example. And finally, we score the structures with statistical potential derived from determined 3D structures from PDB to get the result of RNA folding. So here I will show you a movie. On the left panel, you will see the structure of the coarse-grained RNA. And on the right panel, you will see how the energy changes and the RMSD to the native crystal structure changes. So we want the RMSD to be the lowest, which will mean the structure is the most similar to the reference. So let me play the movie. And you can see that the RNA is flickering, moving around, doing different moves, different pairings. And on the right side, you can see how it explores the energy and structural landscape. And now we can see that it's more or less started to fluctuate in the low RMSD, low energy region, which means that we are able to find and lock the RNA closest to its native state. And this was simulation only for one run simulations, but what we actually do is a replica exchange Monte Carlo, which means that we simulate several runs of the same molecule in different temperatures. And after a specific number of steps of simulations, they exchange between the structure exchange between the temperatures, which allows us to actually hop over the energy barrier. So you can see that the big blob in the middle of the right panel is a huge set of misfolded structures. The one blob on the top is basically from the very beginning of the simulations and from the very high temperature simulations. But by changing the replicas, we are able to lock some of the molecules in the low energy states, which resemble the native structure. And this, so the SimRNA method actually is well performing with replication, the folding funnel for many RNA molecules. And the folding funnel is on the right side is similar to what you've seen on the previous slide. So we search the space and we want to get as close to native as possible. And we are quite successful with that with many RNAs. As you can see on the bottom, for all those examples, you can see that the folding funnel is actually getting to the native-like state. And now we are approaching to the point where we actually will be using the LUMI Supercomputer. And as many of you know, or probably all of you know, that more than a year ago, pandemic started. This is caused by SARS-CoV-2 beta coronavirus. This is the SARS-CoV-2 beta coronavirus is one of the largest RNA viruses with around 30,000 nucleotides. And of course, this is very difficult to study. So at the beginning of last year, Professor Dani Incarnato approached us and asked us for help with his studies, where he was probing the SARS-CoV-2 genome to derive its secondary structure, and asked us for help to actually work on the secondary structure and try to model 3D structure of some of the fragments of SARS-CoV-2 virus. And actually, this resulted in a very good publication where we were the first group of scientists in the world that actually were able to define the whole secondary structure of the SARS-CoV-2 virus. So here you can see parts of the secondary structure of the virus. They are derived by chemical probing and predictions. You can see it has very, very interesting structures throughout the span of the whole genome. And then this was a huge work, and following the secondary structure predictions, we focused on the most conserved among beta coronavirus family structures to further study them in 3D and find potential druggable pockets in them. So for those, we run SimRNA modeling with restraints from the secondary structure simulations. We got the whole ensemble of structures for each of the 9 fragments that we choose. Then we use the software called fPocket, which is created for finding druggable pockets or ligand binding pockets in proteins, but we tweaked it a little bit, benchmarked, and use it for finding pockets in RNA. And we were able to specify from 1 to several pockets in each of the studied fragments of the SARS-CoV-2 virus that we simulated. And here, for example, you can see how the pockets look like. So the pocket 1 is in a multi-way junction, and you can see that it's like a nice hole in a structure where you can imagine that the ligand comes inside and possibly locks this structure in one state, leaving virus not being able to replicate, for example. Or here on the pocket 2, there is a 3-way junction, and again there is this nice hole where the ligand can fit in. And now I will ask my friend Filip Stefaniak to say a few words how actually we want to simulate the ligands binding to those RNA on the LUMI supercomputer.
Okay, thank you. Can you hear me?
Yes.
Yeah, thank you, Tomasz. So at this stage, we have structures of RNAs, which are potential targets for small molecule ligands. And here, a project which we are going to realize on the LUMI supercomputer. And here we are going to use 3 main components, 3 main technologies. So the first and the second technology we are going to use is molecular docking combined with scoring function. And here, molecular docking, we are using the standard approach, which is slightly optimized in our laboratory, but it is combined with a unique scoring function which is specially designed for RNA and small molecules. And it is based on the AnnapuRNA scoring function developed in our lab recently and published recently. And this scoring function has quite unique features. So first of all, it's use the coarse grain representation of both interacting partners, both RNA and small molecules. And this makes this models we are using here quite universal and general. And we also use machine learning to develop the scoring models. So we believe, and it was checked in the benchmarks, that it's by using these two combined approach, we are using we are getting method which is better than any other existing method for docking and scoring small molecules ligands with RNA. So these are two first technology, and Tomasz, please proceed to the next slide. And the second technology is the extension, and then third technology we are going to use here is the extension of the SimRNA program Tomasz mentioned earlier. And SimRNA program itself is used for folding RNA itself. And here we have the working prototype of SimRNA L program, which extends the capabilities of SimRNA program to fold RNA together with small molecules. So as it is shown in this animation, we are going to use dynamically predict the interactions of small molecules with RNA. And please proceed to the next slide. Thank you. So here's a pipeline and goals of the project we are going to conduct. So first, we are going to find small molecules which potentially binds to the selected RNAs of SARS-CoV-2, and this will be the multi-stage process. So first, we will use the molecular docking of the huge library of hundreds, thousands of compounds. Then we'll use slightly modified scoring functions, which will give us a list of most promising compounds with potential high binding affinity toward selected RNAs. And then for selected top scoring compounds, we will use SimRNA L approach to test if those molecules actually lock or disrupt the RNA of interest. And this large-scale analysis would not be possible without access to resources of the LUMI supercomputer, because all these stages requires a lot of CPU hours to accomplish. And of course, this is not the whole project. We are going to continue this analysis, and for the selected potentially best molecules, we are going to test it if they indeed binds to the selected RNAs, and moreover, if yes, if they are disrupt the structure or lock the structures of the given RNA. And for selected best compounds, we are going to check if they are influenced indeed the virus replication in cells. And the last part of this project will be conducted, of course, outside the LUMI infrastructure. And with that, we would like to thank all current and past members of Bujnicki lab, and also our collaborators, Dani, Martin, Sebastian, Zaoming, Witold, and Matthias for their help in this project, and the financials body and you for your attentions. Thank you.
Thank you.
Thank you very much. We have one question waiting in the Q&A session. How do you think it is possible to obtain these same secondary RNA structures using all atom force field in water?
So secondary structure is separate from 3D structure, but if we are talking about all atom representation in water, I would assume that we are talking about 3D structure, right? Am I correct?
We need to assume, we have something, yes.
Okay, so all atom simulations in in a 3D with explicit water, they are much more. I don't want to give a numbers, but about 1,000 times more computationally demanding that the simulations that we do in coarse grains representation, and actually our force field, our method is not capable of running all atom simulations at this point. So I believe that we would be able to get similar or better results using all atom representation, but then first we will need to have such methods, and for now we do not have such a method, and then we would need to use the much more supercomputing power and hours to get to the point where we can get with our coarse grained representation in much faster time.
Okay. The question is whether LUMI is not enough.
It's probably it is enough, but I don't know for how many months we would need to occupy it. That is one thing, and the other, actually, the limitation is here that we do not have such a method. I mean, we as a group we don't have such a method, and we are mostly involved in running Monte Carlo simulations. There are methods for molecular dynamics simulations like GROMACS or others, where you can actually try to simulate it, but this is not the field of our lab. So basically, if there is a lab that has an idea and knows how to run those simulations and want to use LUMI Supercomputer, then probably it would be enough. But I cannot tell in the time how much time it would take to get to those structures.
Okay, thank you very much from the person asking the question, and also thank you very much from my side. That was a great presentation, and I think that both presentations actually show that the need of expansion of the HPC resources in Poland and in science in general is growing, and it actually has become a very basic tool for various fields of science. And I hope that LUMI will be a good step in providing those high-end capability resources for the needs of Polish groups, in parallel to the EuroHPC efforts on the European level, of course.